Okay, so this is part 2 of section 4.0, which is action potentials and synapses. So we've already covered the action potentials. So now we're going to talk about what the synapses is. And so let's get started here. So first, let's say, let's start, get a pen here. So we're going to be talking about the synapses. Okay. So just some general anatomy of a synapses. So if we have this sort of generic neuron, right? And has a cell body, as an axon, then down at the end, we have this axon terminal that we've talked about. Okay. And if we expand this a little bit more, we can say we have something that looks kind of like this. Okay. And so what you see here is basically going to be another cell body, axon. So we're propagating action potential down the axon to the terminal and then across to the next neuron. So we'll say that the axon. So if this neuron right here is the first one is propagating the signal and transferring it to the next one, we would call this a presynaptic side here. Okay, so if we're on this side right here, this is the presynaptic. Okay, and then this would be the post. So this here would be presynaptic. This would be post synapse. Okay. This here would be the axon. Okay. And signal would be coming down this way, the action potential, and it would reach the axon terminal. Okay, talked about that before. So we have the axon terminal. We have this space here between the pre and post synapses, and this is what we call the synaptic cleft. So we'll talk about how when we transmit a signal, we will basically release a neurotransmitter into the cleft. Okay. And there will be some sort of receptor, okay, on the postsynaptic postganglionic neuron. If this is another neuron, or the postsynaptic side will have some sort of channel. It has a receptor, and that neurotransmitter will hit the receptor, and it will sort of open up the channel, and it will, if it is a sodium channel, let's say, then we will be getting sodium that is in the synaptic cleft, and it'll be rushing into into the postsynaptic neuron, or dendrite, we'll say, and it will be ultimately be this would be voltage gated, right? So we have electrochemical gradients we discussed. So this would be generating a graded potential, right? So it'd be stimulating this postsynaptic neuron here, this one, and if it reached the -55 threshold, then we would get a action potential that would be propagated down its axon, and it could interact with another neuron, or it could interact with an effector cell like a skeletal muscle, or at a neuromuscular junction or something like that. So that's just sort of some like basic anatomy of the synapses. Okay. Another thing we could talk about here would be we could say that let's talk about a couple different kinds of synapses here. So we have synapses. We're going to talk about a chemical synapses and an electrical synapses. Okay, and we'll say that a chemical synapses relies on a neurotransmitter, and we'll say that an electrical synapses is basically a presynaptic and postsynaptic membranes, okay, that are joined. So we'll say pre and post synapses that are joined. Okay, and we'll talk about all this more. So a chemical synapses is dependent on like a neurotransmitter. And an example of this, we could talk about, let's do acetylcholine. And we call this so acetylcholine, we call a cholinergic synapses. Okay, if we go back to that that sort of generic synapses we drew out before, and we can draw out another one here and talk about it more in depth. So if we're talking about a cholinergic synapses, we have a lot of things that are going on in the axon terminal. Okay, the axon terminal has a lot of enzymes, and it has a lot of synthesis going on, a lot of transmission. So we have synthesis, we have transmission. Okay, we have packaging, we have recycling. Okay, these are like a lot of just general functions that are going on in the axon terminal. And just to sort of describe like this process that's involved, sort of a high level, what the process involved with acetylcholine. Let's get another color here. So let's take this A, okay, and then let's take CH. Okay. So there's a process going on here to make acetylcholine. You have acetyl and you have choline that are converged together. Okay, and those make acetylcholine. And to get them, so you'll have sources of this, right? If you go to the mitochondria, mitochondria is a source of acid. So acetyl coenzyme A. Acetyl coenzyme A, a source from the mitochondria, and this can be a source for your acetyl, okay, and it can be broken in acetyl CoA can be broken into acetyl and into CoA, and then recycle back into mitochondria. So what can happen then is you get acetyl CoA, you break off the CoA, send it back to the mitochondria, and then you attach the acetyl to a choline molecule, and then these two become acetylcholine. Okay, this acetylcholine, okay, is it is packaged, all right, into these things we call vesicles. Okay, so you have a lot of this acetylcholine packaged into vesicles. These vesicles are sort of hanging out here in the axon terminal, and then they can sort of come and merge, okay, merge with the membrane here and be released into the synaptic cleft, okay. And what would end up happening is that you would have, like we mentioned just a few minutes ago, you would have this this receptor, this channel here, right? So this channel is going to first be closed, and let's say that this channel is sodium channel, okay? And it is first closed, but then once that acetylcholine hits the acetylcholine space on this channel, right? It hits a receptor there. Then what'll end up happening is the sodium will end up coming into the postsynaptic through the postsynaptic membrane through the cleft, okay? Because sodium is sort of hanging out in here as well. And then what we can do is we need to take this this remaining acetylcholine, right, that is within the cleft. It's filled up in the space here, and we need to take this acetylcholine, and then we need to break it back into acetyl and into choline, right? So we need to break it back into some of its constituent molecules here, and then we need to sort of recycle, okay, recycle this choline. And the way that we are able to do that is we have this molecule here that is called acetylcholine esterase, and this protein here is esterase protein, is specific to work with acetylcholine, and what it does is it as esterases do, so they will basically just take this acetylcholine molecule, break it in half, right? And then they will recycle, then the choline will be able to be recycled, taken back into the presynaptic terminal, and then conjoined with another piece of acetyl and to form a new acetylcholine molecule, and then sort of cycle continues, okay? Just a quick note as well. So we also have, we're not going to talk about it in this part of the lecture, should be another time, but there is an importance of bringing in calcium, okay? So you will see a lot of calcium channels, okay, on very abundance of calcium channels on the presynaptic terminals, and because calcium comes in and it is responsible for actually pushing these vesicles down to the terminal to release. So they're very the calcium is important for release of a neurotransmitter, and in this case it's acetylcholine. Okay, so this is an example of a chemical synapses, and I just want to hit a couple points here too. We have something that is called, let's write it down here. Okay, so it is called fatigue. Okay, so it's called synaptic fatigue. And this is basically when the rate of a neurotransmitter releases, well, the rate of the neurotransmitter release. Okay, so that's this part right here, the rate of the release surpasses the rate of the recycle. So that means that you are dumping more neurotransmitter into the cleft, right? And when that rate surpasses the recycling capability, then we're going to end up having fatigue. So what we end up with is basically a dysfunctional axon terminal. Okay, sort of exhausts out the other neuron. It doesn't give it a chance to reset and to reset and have another action potential started. Then we have something here. Let's draw it over here. Actually, we have something called synaptic delay, and actually it's a delay. Okay, so we have fatigue and we have delay. All right, talked about fatigue. So now delay. This is basically the time it takes, okay, to propagate a signal down here and release some acetylcholine or a neurotransmitter of any kind into the cleft, cross over the cleft, and hit a receptor, and then causing the ion, whatever it is, the ion to sort of come in and then initiate another action potential, right, along its axon. Then the time that it takes to do that, okay, we'll say is somewhere around 0.2 to 0.5 milliseconds. Okay, and this is a long time. If you are considering that, like maybe you're stepping on a nail or you're placing your hand on something hot, you need a quick reflex, right? So there's a delay that's happening here, and this is sort of the principle of us having reflex arcs. Okay. So also the time that it takes to, if you sort of collect or get the net sum of all the delay together that it takes to go from the spinal segment where you're bringing that sensation of maybe pain in, and sending it all the way up to the cortex and bouncing around and then back down through the motor tract. You get the net amount of delay there. It could be you know, it can take a while, so we don't want to do that when we have a painful stimulus, right? So that's a principle behind our reflex arch. And I'd also say that our electrical synapses here now, this isn't as much of a big concept here, but the only way that we can get rid of a delay here is that if we have a synapses that maybe is directly coupled. Okay, so if we have a synapses that is directly coupled, where the pre and post synapses are touching each other, they're joined, and in this case we have gap junctions that are here for communication. And when you are able to have this sort of setup right here, okay, you're directly communicating and passing that electrical stimulus, okay, and you are also avoiding the delay. Okay, so one way to get around synaptic delay is to have this electrical or direct coupling. Okay. We have another concept now. Let's talk about here, get our board out. And so here we're going to talk about 4.0 4.8, we're going to talk about these postsynaptic potentials. Okay, postsynaptic potentials. Here we're going to talk about something that we say is an EPSP, or what we call an excitatory. Call it a excitatory postsynaptic potential. Excitatory postsynaptic potential, EPSP. And we're going to talk about an IPSP, okay, which is an inhibitory postsynaptic potential, okay. And this is just really an easy concept, okay. So a excitatory postsynaptic potential is something that remember we have the 0 millivolts, the negative 55, the negative 70, right? So you have the resting membrane potential, it's the threshold, and then we have action potential, then positive 30, and then so this is our depolarization, this is our repolarization, right? Well, we say that an excitatory postsynaptic potential, okay, this is post, meaning we're in the postsynaptic. This is something, this is going to be a potential energy that is excitatory, that is bringing that postsynapsis membrane potential towards threshold. So we are going this direction, okay, so we're going more positive. All right, and we're moving towards threshold to initiate an action potential, right? An inhibitory postsynaptic potential is going to be different. And an inhibitory one is, we're going to say that we're moving away from threshold, so we're moving more negative, okay? And we'll say that a inhibitory postsynaptic potential is going to require a higher grade stimulus to bring the membrane to a threshold, okay? And that's true if you think about it. Let's say, let's get another color here. Let's say that an inhibitory postsynaptic potential is making us more negative, right? Let's say it makes us down to negative 85, okay? So it brings the membrane potential down to negative 85. And in the previous case, from a resting membrane potential, the difference here was 15 millivolts from 70 to 55 for us to be able to get to threshold. Now we have gone even more polarized, and now we have a total of, between this whole space from here to threshold, we now have 30 millivolts. Okay, so we actually have twice the distance, or twice the amount of voltage going in the positive direction that we now need to get to be able to initiate that action potential. So that's what I mean when I say that that a graded hyperpolarization, meaning we're going hyperpolar, right? So this is polar, we're going more polar. So graded hyperpolarization in this negative direction, inhibiting the membrane because it requires a higher graded stimulus, like 30 milli, twice as much in this case, to bring that membrane potential to action potential. Okay. So that's what I mean by this. So we have excitatory and inhibitory signals. We have a concept that we call summation, okay? And when we talk about summation, we say that this that it is the integration of the effects of multiple graded potentials to have a net effect on the membrane potential. Okay, so if we want to look at that generic neuron, right? So we have the neurocell body, then we have the see the axon, and then the axon terminal here. Okay, and so what we say is this is the effects of multiple graded potentials. Okay, multiple graded potentials that have a net, right, a summation, right, effect on the membrane potential. So let's look at that. What is it? What do I mean by that? So let's say that we have some neurons out here, right? There's some neurons that are sending their axons, and they are communicating with this neuron. Okay, and each one of these are sending, let's say, 0.5 and 0.5, right? So I say that we have a net effect of multiple, right? Multiple graded potentials, multiple potentials have a net effect. So this cell body is not just getting 0.5 millivolts from each independently. It's the net effect of the two together. So these two would be 0.5 plus, right? Plus 0.5 would equal 1 millivolt. So the net effect of these multiple graded potentials hitting the cell body is actually 1 millivolt, even though they're 0.5 each. And so that's what I mean when I said that. And then we can further subdivide, okay, temporal. We can further subdivide summation into temporal and into spatial, okay? And temporal means time, okay? And spatial means space, in space. So in space and in time, okay? So an example of a temporal, right? So let's define temporal summation. So a temporal summation is a series of small graded potentials that individually do not generate threshold potential, but due to a rapid rate of firing, the sum of the individual firing generates a greater depolarization. Okay, so what do I mean by that? So what I'm saying is that let's take, for instance, our image up here, right, of our action potential, and let's say we have negative 55, negative 70, right? And let's say that we are sending, we're doing a graded potential, okay? And let's say over this period, we have 0.5, 0.5, 0.5. We have three graded potentials, multiple graded potentials, okay? And the sum of these three, okay, is going to be 1.5 millivolts, right? So 3 times 0.5, 1.5. And the sum of the effect of these three is going to be 1.5 away from 70. Okay, so that's gonna make this point here, the peak of this graded potential, is going to be 68.5. Okay, 68.5. I just messed that up. Let's draw it down here. 68.5. And if we say then that the going back to the statement that I made. So we have a series of small graded potentials that individually, right, 0.5, do not generate an action potential because we did not get to the negative 55 up here. We only got to 68.5. Do not generate a threshold potential. So that's what we explained here. Now let's do the other part of it here. The other part I said is that, but due to a rapid rate, so we'll say a high rate, okay, but due to a high rate of firing, say firing, the sum, okay, the sum of the individual firing generates a greater depolarization, okay. And so what that would look like then here is that if on the next one, all right, let's say we come to this point right here and then back down, all right. So in this case, let's say that we have 0.5, 0.5, 0.5, 0.5, let's say some other ones in here, 1, 2, okay. Now, we take the sum, okay. Let's take the sum of all these, 0.5, 0.5, so this is 2, 4, 5. So the sum of all these are equal to 5, okay? 5 from 70 is equal to 65. Still not negative 55, but it's negative 65, okay? So we had a greater depolarization, okay? When we summed up these, we had a greater depolarization, all right? And then let's just, to be accurate with our diagrams here, let me change one thing, okay? If I can, with my pen. Yep. So to be accurate here, let's say now that this depolarization, okay, happens much quicker, okay? So we're saying just this time here, when previously, let's say the previous one took this much time, right? We'll say this one took this much time. So let's say the rate, so we'll say that the time of each one of these, this one is much faster, so less time. And we also had a much higher rate of firing, right? Which gave us a much higher depolarization, right? 5 as opposed to 6.8. So that's what we're saying here. So we're saying then that with temporal summation, we can have a greater depolarization with a higher rate of firing when we sum up the action potentials, okay? And then with the let's say with this, the spatial synapses, so this is sort of, so let's go back to here. So spatial would be like you have more neurons coming in, right? And these are all sending synapse, right, into the cell body here to communicate, right? Let's say they're all around, right? So sort of encirculating this neuron, right? And you have all these synapses coming into this cell body, okay? And if you sum up the spatial area of these neurons that are sending basically multiple synapses that are active, okay, that are working on that effector or that neuron at so at the same time, Okay, you can sum all these up, and then these will also be able to generate a greater depolarization. Okay, so that's that'll sort of wrap up 4.8 and the discussion on these postsynaptic potentials and summation of charges. Okay. Next, let's talk about let's go ahead now and let's finish off and let's talk about neurotransmitters here. So this will be the last section here. This will be section 4.8. We're talking about neurotransmitters. And what we can do here is we can break up our neurotransmitters into two components or two divisions. Okay, and we're going to do acetylcholine. Okay, and we're going to do what we call G protein. Okay, so we have two divisions, and we talked a little bit already now about acetylcholine. And let's first just say what a neurotransmitter is. Okay, a neurotransmitter, I'd say, is going to be this is going to be a signaling molecule. Okay, and it's secreted from a neuron to some sort of effect, to affect some other cell, okay, or across some sort of synapses, right? So we looked at all that, but it is a signaling molecule, okay? And we can also say that there's this general rule that neurotransmitters, or at least all the selected neurotransmitters that we're going to talk about here, they all have actions in, so we say all, okay, have actions in the central nervous system. And then I would also say that few of them have actions in the peripheral nervous system. And we'll look at some examples of acetylcholine and norepinephrine. Okay, so that's sort of like a general rule. Okay, and let's talk about something that we call a regulatory neuron. And we're not going to go into depth on a regulatory neuron, but if you look at this, I just want you to be aware the sort of generic neuron that we have here, right? So we're sending an action potential. We have these regulatory neurons that are going to have the ability to work on on either helping to like facilitate this action potential across to another neuron, right? So it either be positive, right? Go positive, or it can have a negative or inhibitory effect on this transmission. And that's all I want you to know about what a regulatory neuron is now. Let's clear this out here, and give us a little bit of space again. Next, we want to talk about these G proteins that we mentioned up here, right? So we have these G proteins, and we talked a lot about how acetylcholine was made in the presynaptic axon terminal, right? And then how acetylcholine was sort of synthesized on a high level, and then propagating the signal over to the postsynaptic neuron, right? G proteins are a little bit different. They're extremely high yield as well. So let's take a look at. We're not going to go really in depth into it, but I want you to have a general understanding of what these are. Okay, so G protein. So let's do this sort of generic looking protein. Okay, here we have a membrane. All right, so this is our membrane here, and then we have so we'll call this the extracellular fluid, and then the cytosol. All right, so outside of the cell, inside of the cell. Within the membrane here, we have this G protein, okay, and it's embedded in the membrane. And then this G protein, right, has a receptor on it, and we call that a. Let's go ahead and just name all these out here. So we have a G protein receptor and a G protein. Okay, so the G protein is embedded in there. The receptor is on the extracellular fluid side, on the outside. And what we're going to say is that G proteins are molecular switches. Okay, so we're going to say a G protein is a molecular switch. It's involved in the transmission of signals from outside of the cell into the cell. Okay, and it's activated through G protein receptors, and these receptors on G proteins are going to interact. Okay, they're going to interact with some sort of ligand, right? So we could say a ligand or a a neurotransmitter, right? So they will react with the G protein receptor. They will activate the G protein, and then what we will have is another protein on the membrane close to it. Okay, sort of adjacent to it. And once this G protein is active, it'll basically transmit that signal over to this other enzyme or protein, right? This other enzyme. And in this scenario, we're not going to do in-depth discussion on this, but this enzyme we'll call adenylyl cyclase. Okay, adenylyl cyclase, and it will get it'll get turned on by the G protein, okay, and then it will release another molecule that we will call cyclic AMP, okay, and the cyclic AMP will then go and get let's say it will get activated, or let's say that it'll let's say protein kinase A. Okay, protein kinase A will then trigger just some sort of cellular response. Okay, and a good example of like a cellular response that you would have with like a protein kinase A is going to be something like increasing calcium in your heart. Okay, you increase calcium in the cardiac tissues. It would be like the cardiac muscle. Okay, so that would be like a cellular response inside of the cell. And in order to be able to get this signal of this neurotransmitter extracellularly, it goes through these steps. And what we call this initial signaling molecule, because remember we call neurotransmitters here signaling molecules, right? We call that this acetylcholine is the first messenger. Okay, first messenger. All right, and first messenger is extracellular, and it's going to react to the GPR on the GP. It's going to GP is going to transfer the signal over to the adenylyl cyclase. Adenylyl cyclase is going to release this cyclic AMP, and we're going to call this cyclic AMP another signaling molecule, okay, but it is an intracellular signaling molecule, and we're going to call this the second messenger. And so the second messenger is then going to trigger the remaining part of the pathway to ultimately give us a cellular response, like increase in calcium in the heart. And these two, so the G protein and the messenger system, we actually have a name for that, and that is the G protein linked messenger system. Okay, so this is a G protein linked messenger system is what we refer to when we do this sort of signal transduction. Okay, and let's talk about a few neurotransmitters. Okay, let's go with, so first, since we just talked about the G protein, let's go ahead and talk about a few G protein. Stay on target here. Out of the G protein receptors, we say G protein linked second messenger. We have some that are called norepinephrine and epinephrine. Okay, let's do the norepinephrine and epinephrine. Okay, these are basically adrenaline. Adrenaline is adrenaline and noradrenaline is what it's called. And I'll just do a positive and negative for either excitatory and inhibitory. So these are excitatory. Okay, norepinephrine, epinephrine are excitatory. Then I want you to know, so let's talk a little bit about serotonin. Serotonin, which is also 5-HT, and serotonin is, so this is going to be connected with mood, okay, and we have one called glutamate. Glutamate is very, very, very excitatory. Okay, it's a highly excitatory. And finally, we have something that we call GABA, and the name for that is gamma aminobutyric acid, and this is a very inhibitory, okay, very inhibitory neurotransmitter. And we have a whole drug class called GABA inhibitors. We have glutamate. So this is something that would be responsive as excitatory. We see this in like Huntington's disease. So it's, behind what we call spastic chorea movements. Serotonin is involved with your mood. So drugs like ecstasy, so these deplete your serotonin and they cause a depressed mood, right? Adrenaline, so your norepinephrine and epinephrine, so these are excitatory, and you'll typically find, so you'll find norepinephrine and epinephrine in the peripheral nervous system. Okay, these are going to be at like postganglionic and adrenal. So in the medulla of the adrenal glands, and their synapses. Okay, and these things are going to be affecting like so norepinephrine will say smooth muscle, okay, glandular tissue, and cardiac tissue. very common cardiac tissue. So these are some examples. Okay, and these are all using the G protein linked messenger system. Okay, so all these are G proteins. We're not going to go into depth on each one of these. I just wanted you to just know a little bit of information about each one of them. And then finally, to wrap this up here, let's talk about acetylcholine here. So acetylcholine, and so this is, I would say, this is the most predominant neurotransmitter. So it's the most predominant neurotransmitter in the CNS. Okay. Another thing I'll say about acetylcholine is that it can be found, okay, can be found as well in the peripheral nervous system. And then these are going to be in glandular tissue as well. Okay, it's also going to be in more so in the so we'll say pre-ganglion. Okay, the pre-ganglion. And so we say G proteins are post-ganglionic, can be found. Acetylcholine you'll find pre-ganglionic. Okay, and also you can find this at neuromuscular junction and those sort of synapses as well. Okay, then I think we have a space. Let me create a little bit of a space here. Just get rid of this stuff. Okay. Now, the final statement I want to make here is that that your acetylcholine, these are chemically gated, right, or ligand gated ion channels that we use, right? And an example of these, okay, example are going to be we call nicotinic. When we're referring to acetylcholine, we can call them cholinergic, or we can refer the synapses, okay, and or say the acetylcholine receptors actually. So when talking about the receptors, we refer to them as nicotinic, okay, and the nicotinic receptors we have. So we call here NN, okay, and these are these nicotinic receptors are allowing we'll say an efflux of potassium and an influx of sodium ions. Okay, so whenever you have a nicotinic receptor, they're involved in either potassium going out or sodium going in. Okay, and then we have another type that we have called an M. And M is the same receptor that you would find at the neuromuscular junction. Okay, so if you have that going to the skeletal muscle, you have that action potential. So you have a signal coming from the brain, going down a motor tract out to the periphery, right? And then you have some sort of muscle, right? As a bicep muscle or whatever. And it is stimulating that muscle. And we have what's called a motor end plate, and that synapse that is involved, we'll talk about in more detail in the musculoskeletal. But this here is the neuromuscular junction, okay? And so that is out on the effector scale, and you will find the acetylcholine is being used here. That is the neurotransmitter, and that is the nicotinic with the little M subscript there. Okay, and these are the two subtypes, okay, of the nicotinic receptors, and that's about it. That's all I want to discuss here on this lecture, and we'll pick back up on the next section, and we will be discussing neurophysiology. But I think we will start to talk about some of the brain structures, like the brain stem, and some of the limbic system, the diencephalon, and probably the cerebellum and cerebrum. So we'll sort of look at them individually. Okay, thank you.